Hello, and welcome to part 3 of our lab skills series, Microscope Use and Cell Counting. Brought to you by California North State University, College of Pharmacy. The first thing you need to know before starting to count your cells is how to use the microscope. Begin by locating the green power switch on the microscope base and flipping it to the on position. When your sample is prepared, you will place it on the stand and use the large outer knob to bring the cells into view. The smaller, fine focus knob can then be used to clarify the field. And the small knob below the power switch can be used to adjust the brightness for better visualization. We will be using a hemocytometer to count the number of cells in a cell suspension. The hemocytometer is a modified and calibrated microscope slide designated to allow operators to quickly estimate the concentration of cells in a sample. The cells present in a known volume are counted, and then this value is converted to a number per milliliter. The name refers to its original use in counting blood cells. Blood was diluted to a point where the cells could be reliably counted, and this was factored up based on the dilution. Trypan blue is used when counting cells to indicate the ratio of live to dead cells. The stain turns dead cells blue, but healthy cells with an intact membrane will remain white or colorless. To count cells, add a 10 microliter aliquot of your cell suspension to a clean 1.5 milliliter centrifuge tube. Add an equal volume of Trypan blue and mix well with your pipette. Load 10 microliters of this cell mixture onto the hemocytometer by resting the pipette tip in the valley formed by the cover slip and slowly releasing the mixture from the pipette. Capillary action will draw the liquid in. When you look in the microscope, you will see a field that looks like this, a series of grids and cells. We will only count the cells in the 4 corner quadrants. These quadrants consist of a 4x4 grid or 16 smaller squares. Count the total number of live and dead cells in each of the 4 large quadrants. If you remember, earlier we mentioned that Trypan blue will stain permeable membranes a darker blue. The dark blue cells are dead. Once you have the total number of live and dead cells for each quadrant, add them together for an approximate cell viability calculation. In this example, you can see that 40 of the 47 total cells counted were alive, giving a viability of 0.851 or 85.1%. To find the total cell count, use the following calculation. Step 1. Take the total number of cells counted and divide by the number of quadrants counted. This should be either 4 or 8, depending on if you counted both sides of the hemocytometer. Step 2. Multiply by your dilution factor. In this case, we took 10 microliters of sample and 10 microliters of Trypan. This gives a 1:2 dilution, or a dilution factor of 2. Step 3. Multiply this number by 10,000. This will give you your cell concentration in cells per milliliter. Step 4. Multiply the concentration by your suspension volume to get a total cell count. In this case, our volume is 5 milliliters. For this example, we have 1 million cells, or 200,000 cells per milliliter. This cell count can now be used to plate and seed cells at a specific density. Thank you so much for watching. We hope this has been helpful. Please come back for more lab skills videos brought to you by California North State University College of Pharmacy.